metabolomics data for 1,000 of them. It's untargeted metabolomics, and we have LC-MS/MS, so we have information that's quasi-quantitative, and also we have information about the identity of the metabolites. And finally, we also have shallow shotgun sequencing data for the same subset of 1,000 rats. So these are all data that I'm hoping to integrate in the near future. But for now, my talk tonight will really focus mostly on integrating host genotypes and cecal microbiomes. And so as I was saying, this is a collaboration with the NIDA Center for GWAS bred rats, and also with the Center for Microbiome Innovation, which is based at UCSD. So the data collection was the following. Just a quick reminder, the rats were bred by the amazing Leah Solberg-Wood, originally at Medical College of Wisconsin, and then they were sending siblings to the 3 different centers. So something that is quite important for my studies, that at least in theory, there should be 1 male, 1 female from each SIP ship in each of the 3 phenotyping centers, so Michigan, Buffalo, and Tennessee. And here in Tennessee, we have 2 samples of rats. Some of them were phenotyped for behaviors mostly, but also others were breeders. And so all these rats were, sacrificed and samples taken and sent to the University of California at San Diego to get the genotypes, the microbiome, and the metabolome. So one of the first things that I looked at is whether rats have different gut microbiomes in the different phenotyping centers, and also if they have different microbiomes between the young adolescent rats that were phenotyped in Tennessee and the older adult breeders. And so I used the tool Cheetah to do this. It's a really useful tool that's developed by the lab of Rob Knight. And from this picture here, which is sort of a PCA, we can see, and each color represents a study. We can see that clearly the rats cluster by study in terms of their microbiome. So the rats do have distinct gut microbiomes, depending on whether they were phenotyped in Michigan or in Tennessee or in New York. And there's also a distinction actually between Tennessee breeders and Tennessee phenotyped rats. So my idea here was to initially analyze each of the 4 studies separately, and then leverage the fact that we do have the same population that was sent to the 4 different centers, and that I'm actually using the same pipeline to analyze the data later on. So I'll come back to that. I wanted to go through the strengths of this study, and the first one, as I was alluding to before, is that we don't have siblings in the same cage. And that means that genetic effects are not confounded with cage effects. And we know that cage effects can be quite strong in microbiome studies, whereby rats or mice sharing a cage tend to have more similar microbiomes. So if animals sharing a cage were siblings, you can see that we might misinterpret cage effects as genetic effects. It is not the case here, and the plot I'm showing you here gives you the heritability estimated for a lot of different taxa in Michigan and New York, in a model that accounts for cage effects, and in a model that does not account for cage effects. And you can see there's really very little difference. That's because they are not confounded. And then, as I was saying, also something great that we can do here is to compare across studies. First of all, in each study, Michigan, New York, Tennessee breeders, Tennessee phenotyped, actually not the Tennessee breeders, but the other 3, the sample sizes are large enough to run meaningful genetic analyses. We have at least 1,000 rats in each study, and importantly, the rats come from the same population, and of course the same microbiome and metabolome protocols have been applied, and the same computational pipeline as well. So there's really an opportunity here to compare across the studies. Now, whether we call that trying to see if the results replicate or whether the results generalize is a bit of a question. Replication should be the exact same same conditions, and animals, it's not the same because different phenotypes were collected in the 3 centers. So I prefer to talk about generalizability, and that's what we want really. We like our results to be generalizable. And something important too is that we have microbiome, but also metabolome data and hundreds of behavioral and non-behavioral phenotypes for each of the rats, so metabolome for a subset, but phenotypes for each of the rats for which we have microbiome. So there is an opportunity again to put the results that we have for the microbiome in context by comparing, for example, the heritability of the microbiome with the heritability of behavioral and non-behavioral phenotypes. And then, of course, we can do, systems genetics analysis to try to integrate the different levels of data and better understand the biology. And finally, we shouldn't forget this population is highly recombinant, with many recombination events having accumulated over the generations of outbreeding. And so when I studied HS rats for the first time during my PhD already, we had a good chance of identifying, putatively causal genes at QTLs. I estimated that at about 1/10 of the QTLs, we had a good idea of what might be the causal gene. It can only be better now because there's been more generations and more recombination. So we do really have a population that permits high precision mapping. So the first question that I've addressed is the microbiome heritable? And so this is a very important question in its own right, and there's been a lot of debate around this. So for example, I'm showing you here 2 different papers, one from humans that was saying that really there's very little host genetic effects on the microbiome. It's all about the environment. And another paper that came out recently from wild baboons saying all microbial taxa are heritable, and both papers are remarkable. The claims are really well supported by the data and results. So there's my thought about it was that for the microbial traits, just like for other phenotypes, it's likely that there is a genetic basis to them, which can be small, but if we get enough data, if we have enough statistical power, we can detect heritable, the heritability, the genetic basis of the trait. So I'll revisit that with the data. Because we can compare across studies, it's interesting to ask whether the taxa heritabilities are correlated in 2 different centers. And also, how does microbial heritability compare to organismal phenotypes? Because we need to remember that heritability is population specific. For example, if you get a population with recombinant inbred strains and you get 100 replicates per strain, you will have a very, very strong heritability, but that doesn't mean much in itself. I think what's really interesting is to compare with other phenotypes collected in the same population and animals in that case. So first question, is the microbiome heritable? There's multiple ways to sort of address that question. The one that I prefer, I think, is to show QQ plots. So these QQ plots have on one axis, on the x-axis, the expected p-value of the test heritability is equal to 0 or different from 0. And on the y-axis, we have the observed p-values for that same test. And so here, each dot is a microbial taxa. The 4 quadrants here correspond to the 4 different studies. So the null hypothesis, again, is that the microbial taxa are not heritable. And if we see that there's inflation, meaning the dots are above the diagonal, then that indicates that the taxa tend to be heritable. And we can see here that in 3 quadrants, there's clear inflation of the p-values. That is not the case in Tennessee breeders. So there is evidence in those 3, not in this one, but we need to remember that the Tennessee breeders, the sample size is half of the other ones. And so there is no evidence, but it's possible that it's due to a lack of statistical power. So there is evidence the microbiome is heritable across the board of microbial taxa. Now, are the heritabilities correlated across studies? So in each plot here, I have the heritability for microbial taxa that was observed in the two studies. So the first row, we have New York, Michigan here, Tennessee behavior, Tennessee breeders. So for example, if we look at this plot, and I have to say all the axes have the same scale, of course, we tend to see some little correlation in that quadrant here. And indeed, there is a correlation that is significantly different from zero, even after accounting for the 6 tests that we're doing here. So between New York and, well, in New York and Tennessee behavior, the heritabilities tend to be correlated across taxa. So if a taxa is highly heritable in New York, it tends to also be quite highly heritable in Tennessee behavior. But overall, really, there's still little evidence that the heritabilities are correlated. So that could mean that truly there's no correlation or little correlation, or it could be that our estimates are too imprecise and we can't really see the correlation that exists. And so keeping in mind that heritability estimates are population specific, what makes the most sense to me to do is to compare the microbial taxa to organismal phenotypes. And I've split here between behavioral and non-behavioral phenotypes, because we know that behaviors tend to have lower heritabilities than non-behavioral phenotypes. And what we can see on this plot is that the microbial traits, which are in some shade of gray, are indeed have lower heritabilities in HS rats than behavioral and non-behavioral phenotypes. And we do see that behavioral phenotypes, well, so interesting. I'm sorry, I think there might be sort of a swap of labels. I'm pretty sure that the behavioral are less heritable than non-behavioral. So coming back to the 2 papers that were published before, heritability of the microbiome is quite low, but if we have enough data, we can detect it. We can see that it's significant.
Amelie, are you
wrapping
up? 1 more minute?
Yeah.
Running a bit out of time.
Yes, I'll wrap up now.
Thank you.
Yeah. So I've been talking about genetic effects across the genome, heritability. Now we are also looking at QTLs and trying to map genetic effects. And something that I'm really interested in doing is identifying QTLs that are observed in all 4 studies, as is the case in this one. And then, of course, we want to integrate the phenotypes and do mediation analysis like Leah presented before. So I definitely need to up my game on that. So I've already acknowledged the NIDA Center, the Center for Microbiome Innovation, and then there's many individuals who are collaborators as part of this. And I just want to say that I'm looking for a postdoc to work with me at the Center for Genomic Regulation on host microbiome interactions, or potentially on indirect genetic effects, which is the other thing I do. So if you might be interested, you can check out my latest paper, and that will give you an idea of what I do on indirect genetic effects. And I'll take questions if we have time.